smaller grain sizes mean really sandy sediment, and larger grain sizes, higher percent mud, means really fine sediment. And you can see there's a really nice relationship between total organic material and grain size. So as the grains get smaller, you can store more carbon in your sediment. And then after you get to really fine grain sizes, it's so fine we can't even sit it, and it falls apart, and you just see an increase in total organic material at the 100% mud data. So there's a mechanism that is associated with this. There's just increased oxygen availability in sandier sediments such that you can flush water through those sediments more effectively, which oxygenates the sediments and allows increased degradation of organic material. There's been some proposal to use grain size to measure in a stock assessment framework, and you could do that, but what these data say is that basically, unless you have sandy sediments, I wouldn't do that. So in this case, once you get to about 82% mud, the relationship falls apart. It's expensive and costly to go out and measure carbon stock assessments. So if you could go out and just measure how sandy the sediment was, you'd be saving a lot of time and money. But you have to be cautious using that kind of an approach if you have really muddy sediments. And just to show you a little bit why our bare sediments didn't store more carbon than our seagrass meadows did, a lot of that is driven by this site in the middle on the bottom here that shows you that the bare sediment in Cypress Grove, which is in Tomales Bay, actually had much finer sediment than the seagrass meadow did. And that's not typical because the seagrass meadows can usually settle out particulate organic material, which leads to finer, muddier sediments inside the meadow, which drives those differences. But we actually had this really anomalous site in Cypress Grove, which I think when you exclude that from all of our bare sediment data, it does show the opposite, such that the seagrasses are actually storing more carbon than the vegetable sediments. But I chose to leave it in because it's still a real trend. So it sort of begs the question, what drives springs on those existing habitats, and there are a lot of different things that can go into that, particularly hydrodynamic and morphology of the estuary that we're working in. There's a lot of recent research being done on what drives those differences that's really interesting and can have huge implications for stock assessments. The last thing that I did was to look at carbon exchange and lateral fluxes between these habitats. So this is the salt marsh that I measured in Tomales Bay, and that line in the bay is seagrass rack. So there's a massive amount of carbon that is absorbed from the sea surface and absorbed from the water column in the seagrass meadow that we talked about earlier on, and contributes to an increase in seagrass biomass in the meadow. But we saw that there's this big seasonal signal between high summer biomass of seagrass and low winter biomass. So where does all that carbon go, essentially, if it's absorbed locally in the seagrass meadow? And a ton of rack gets washed into nearby neighboring habitats. And you can go through historical imagery from satellites and see these persistent rack lines of seagrass in the salt marsh. So I made this biplot here, which is essentially trying to source the carbon in the salt marsh sediment underneath these rack lines. So the question I was trying to answer is the seagrass exported from the meadow, deposited into neighboring areas, and then buried in sediment, or is that just all remineralized and converted back to atmospheric CO2? And those light blue and red circles are the sediment samples. The blue are the sediment samples that came from underneath the seagrass rack. The red are sediment samples that came from elsewhere in the marsh. And you can kind of see that those blue sediment samples are closer up to that seagrass box at the top. So visually, it looks like some of that seagrass may be buried in the sediment, whereas in places where there's no rack, you don't see such a strong seagrass signal in terms of the source of carbon buried in sediment. But when you model that, it's not significant. So this is a figure of the proportion of carbon buried proportion of each source of carbon that's buried in sediment. And the dominant signal in the salt marsh is benthic diatoms or plankton or suspended particulate organic material. They all have a very similar signal. But for the most part, it's small planktonic diatom communities that are contributing to the carbon buried in the salt marsh. And then the next important driver is C3 plants. So pickleweed is the dominant C3 plant in these salt marshes. And then C4 saltgrass and seagrass is not significantly different from zero. So essentially, the carbon that is exported laterally from the seagrass meadows, for the most part, is remineralized offsite. In this particular case, we could be seeing some export of organic material from the seagrass meadow that then is exported to deeper waters and buried elsewhere. But then it's, it's a slippery slope chasing down individual blades of seagrass bed and trying to figure out where they're remineralized or buried. So remineralized here. We also did the same thing in the seagrass meadow, and this is a similar finding essentially. In this case, about 40% of the carbon buried in seagrass meadow sediment was seagrass itself, and 40 was SPOM, phytoplankton, diatoms, similar signal, and then 22% terrestrial in one of these sites. So the take home from this sourcing is kind of that they are important at burying carbon, but the carbon that they bury might not necessarily be their own biomass. They are facilitating the burial of carbon, but by having small grain sizes and low oxygen and inundated environments, they're facilitating carbon burial even if it's not necessarily their own biomass that does it. To summarize all of that information, we know that the seagrass meadows are a net sink annually at the sea surface in terms of gas transfer, 490 grams of carbon per meter per year. They are a net sink in terms of metabolism. I didn't get to show the actual, that's not a metabolic flux, that's just an estimate of pH modification. We've modeled some of the metabolism, but I didn't get a chance to fit it into the slides today, but they are a net sink of carbon in terms of metabolism. And then they also bury and store carbon in their sediment underneath. And there's a ton of questions that I hope to continue to answer in each of these carbon fluxes. But we did find really positive results in terms of their carbon value. And I like to remind everybody that just because they're good at absorbing carbon, they're also good at all kinds of other ecosystem services. So at least from a management perspective, even if in Southern California they don't modify pH in the same way that they do in Northern California, it's still worth restoring or conserving your seagrass meadow. And these are just some of the next questions I'm planning on answering within my postdoc. So what are the drivers of blue carbon storage? I'll be working with someone named Dr. Peter McCready in Australia to look at the actual mechanisms that prevents the degradation of carbon in sediment. And then continuing on this geospatial assessment of OA vulnerability with some people that I've been doing that at the end of my PhD, hopefully adding Ariel Levine into that mix from San Diego State, and trying to understand where hotspots of ocean acidification overlap with ecological and economic hotspots. You can see from that map that there are some areas of California that are particularly vulnerable, but do those areas also overlay with populations of vulnerable species, so shellfish, or human communities, so fisheries communities that might be particularly sensitive. And I would like to acknowledge a whole lot of people and funding sources. Not everyone is on this slide, but it's been a long haul, and now it's over. I'll take questions.
So I know your primary focus is ocean acidification, and that's good because geographical history tells us that can be a very, very dangerous thing. Mm-hmm. But at least where I'm from, when carbon sources get sequestered beneath sediment, highly active anaerobes very quickly convert a lot of that carbon straight to methane, which just gets re-released back into the atmosphere. Have y'all thought about doing some sort of cost-benefit analysis based off of like how much benefit you're getting from removing the carbon dioxide from the ocean waters to versus the amount of methane that actually gets put out into the atmosphere during the sequestration process? Because methane is a fairly potent greenhouse gas, right?
Great question. So in the seagrass meadow, there in this really saline environments, the methane emissions are usually pretty minimal
Okay.
because of the communities and sediment. That's not to say, we didn't measure methane emissions, so there could be trace methane emissions, but they're much less than you would see in a freshwater marsh or even a brackish marsh.
Okay, yeah. That's,
Yeah.
I did all my whole life research in Louisiana.
Yeah.
So I was just like, methane, dangerous.
Yeah.
Yeah.
So I don't know, but we did not measure methane because typically in saline environments, it's not anywhere close to the same extent of emissions.
Makes sense.
Yeah.
Thank you.
In looking at your, first of all, very nice. In looking at kind of the drivers of this, right? You danced around both physical and biological processes that are interacting at the same time. What are your thoughts on the relative importance of physical coupling versus biological coupling in driving variability in these? That's part A, at least.
The ecologist in me wants to say that it's all biological, but I don't think that's true. In fact, I think the physical drivers are actually more important than the biological drivers. I mean, depends on which flux, blah, 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 you're talking about. In terms of air-sea fluxes, I think it's heavily physically and oceanographically dominated. There's definitely a signal in there. I also think that we've underestimated the contribution of water column and phytoplankton productivity in the past for both metabolic fluxes and air-sea fluxes. And from some of Abby's chamber data, we can see that the phytoplankton communities are strong enough to almost reach the same level of productivity that the eelgrass are. And that's really interesting. So it's both, but in terms of the ability for seagrass meadows to ameliorate OA, we've put it all into a model, and biomass didn't even come out as a significant driver. Neither did leaf area index, neither did canopy height. We know it's important, but it's overshadowed statistically by the physical drivers. So hydrodynamics, flow speed, residence time, those drivers are super important because if the water's just ripping through a bed, you don't see the meaningful impact on water chemistry that you do in this slow flow environments.
So having said that, okay, now I'm thinking about, so when you cast your answer in the end, you say, these things they provide this. Given that it's physical coupling and you go along the coast, those physics are going to change hugely as you go, and the way they interact with the microbe and all those things. So what do you think this means to your predictions about geographic variability in the story you just told?
Oh yeah, okay, that's a big question. I mean, in terms of Zoster marina, Southern California is close to its southern range limit. And I think some of the geographic variability we see in the faster timescale processes are driven by stressful conditions in Southern California. We do not see the same ability for the seagrass meadow to modify pH in Southern California that we do in Northern California. And part of that, I think, is that the higher temperatures are facilitating increased respiration. Currents are kind of slower down here, but that's not necessarily true on a bay-wide scale. I think that maybe along the large-scale coastal currents, but I don't know that that's impacting what we're seeing here on this scale. And then the whole OA geospatial part is a whole nother question because we get really corrosive conditions up on the north coast. But we still see corrosive conditions down here, but their drivers like eutrophication and nutrient runoff and sewage outfall are more important in Southern California. So I don't know. There's a whole bunch of variability spatially, but I do think the sort of Northern California, Southern California differences are going to be driven by temperature for Zostera marina, and then the sort of separation of the global acidification impacts in Northern California and the more urban impacts in Southern California. Teresa?
Super cool, thanks for sharing. Unsurprisingly, I'm really interested in like the shellfish management
Mhm.
things that you bring up. So I wonder, like it sounds like the mesocosm experiments you did are really impactful for thinking about oyster management. But then the sort of like restoration benefit timeline that you're talking about means like what are your recommendations, I guess, for helping out existing oyster reefs if restoration is going to take too long to see
Yeah.
short-term like improvements that we need?
So what I would recommend to an oyster grower versus a manager who, let's say an airline wanted to do an offset project and wanted to restore seagrass. Those are different questions because of the time scales that they would be operating on. So the restoration for the seagrass meadow being really, really slow is strictly in terms of the amount of time it would take to sequester in sediment a massive amount of carbon. But the OA amelioration component works instantaneously, assuming you restored a seagrass meadow that was in a location that could provide that service. As soon as it's operating under net autotrophy and a healthy meadow, it could do that. So, like I said, we only did that in the lab, and actually what we see is that an increase in seagrass biomass translates to thicker shell in oysters linearly, and a nonlinear relationship between tissue mass, such that medium levels of seagrass biomass are the best for actual tissue. So if you're a grower, you don't really care if your shell's really thick, if it has less tissue, because that's not what people are eating. So there's work to be done on that and picking apart that stuff. But I actually think that the shellfish management implications are a little bit easier than the sequestration management because of the timescales that you're operating on for those two different things. Chantal.
Great talk.
Thank you.
So in your data slide where you pull all the data showing the Wangkong is more acidic outside of the meadow than it was inside. So my question is, I mean, that's a remarkable observation. My question is there a site that perhaps vastly different compared to others that could explain this difference? Or if you do this comparison side by side, do you still see the same pattern?
Yeah, I think I understand the question. I mean, we have like 25 deployments or something, and in some cases, there's no difference between the seagrass meadow and the outside of the seagrass meadow. And in some cases, it's really big. And those are the cases that we should be looking at to describe mechanisms. And it's been really hard to figure that out. I think a lot of it is driven by residence time and hydrodynamics, where the flow is really fast. We don't get the same ability, but I don't actually know. We don't know the answer to that question still. The number of data points is compelling. So we know it's happening. But yes, we should look to those cases where the difference is really high, and then the cases where it's not different at all. And in some cases that happens in the same meadow at different times. So it's a big, messy data set. Yeah.
I wouldn't back off on the biological importance that quickly.
Yeah.
Part of it, I mean, one thing that the physical does is to act on the biological effects, right? So if, as you pointed out, if you don't have a air-sea gradient, no matter how fast the wind speed is, you're not gonna have flux.
Yeah.
So you can blow 100 miles an hour, you're not gonna have flux. I mean, so part of it is a time scale, right? And so on a short term, you really see the effect of wind on the flux. But again, you have to factor the biological age. And even upwelling, which is a huge signal, has the initial efflux, but then the long-term uptake,
Yeah.
which is, a combination of physical, biological. So
It's true.
I would hang tough on that one.
I guess the better answer would be that they both depend on one another, so you can't answer that question.
How did you determine sedimentation rates?
Some people use feldspar, like a little piece of fabric that you stick in and you measure how much sediment accumulates on top. And that will give you a faster scale. We use lead-210 isotope dating. So it's like a longer time scale sedimentation rate. Lead-210. Any more questions? Oh, Abby?
I'm always struck by the fact it takes over 500 years to get a meter square of sediment, and what the implications that are for the eelgrass mitigation policy. Because, basically, especially in the bay, whenever they need to dredge, they take up all the eelgrass. I saw it happening in Mission Bay during my deployments. They take up all the eelgrass, they dredge, and then they put the eelgrass habitat down. And I understand that's important for, reserving it as a nursery habitat, but basically you're destroying all the sediment. So I don't know if you have any thoughts on that.
Yeah, when the California eelgrass mitigation policy was written, this was all kind of a back burner thing, and the language in that policy, that was based off of a 2007 policy written by Keith Merkel. That was a Southern California specific policy. Then California adopted a policy in 2014 that was statewide, and this was not a part of the ecosystem services that they were thinking about. The language in the policy actually says, no loss or degradation of eelgrass habitat and the services that it provides. But it doesn't really state in specificity all of those things. So you could argue that, but I don't think that this must then therefore go on to sequester blah amount of carbon is a service that people are held accountable for. And this is also only for mitigation, right? If you there are other activities that you're not required to, or they're restoring seagrass meadows, but that aren't thinking about carbon sequestration. So there's work to do, I guess. And I think that we'll see people move in that direction down the road. Todd?
For these different rates you're calculating on different vegetations, have you thought about how potential sea level rise is going to influence these beds, as I would expect seagrass beds are going to be reduced because of lower light penetration, and their metabolic activity and photosynthesis also decline. And when you have the kind of embayments where it's urban, so there's no place for them to really go upwards, right?
Yep.
So what's
Yeah.
any potential impacts that you
It can be both a good thing and a bad thing. In some cases, the sedimentation rates can actually keep pace with sea level rise. So if they had somewhere to go, they'd go there. But they don't always have somewhere to go. So in the cases like in Tomales Bay, Walker Salt Marsh, there's a seagrass meadow, might have a map. There's a seagrass meadow that is right next to, oh, this is my favorite slide, I didn't get to show it. I guess I don't have a map. Well, anyway, there's a seagrass meadow right abutted up next to a salt marsh, which abuts up next to a river and a freshwater marsh. So that's an ideal case, and where the sedimentation rates are keeping pace with sea level rise projections, you can actually see the seagrass meadows, would be moving up, and the salt marsh would be moving back. Great, but in the case of sediment or in shoreline armoring, etc., they'll disappear, and I think that's just what's gonna happen. There may be opportunities. There's all these different levers you can pull management wise. So if they can't march up with the sea level rise, can you improve water quality such that then you're buying them some time? So those kinds of opportunities are there in many different ways. If you can't do X, do Y. So, yeah.
I think we better stop there. Let's thank Melissa again. Bring your remaining questions to lunch or to the reception. Thanks very much.
Sure, thank you, yeah. That's a wrap.